This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 210, recorded on December 7th, 2012. Hi, everyone. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today here in TWiV studio land, Dickson Despommier.
Hello, Vincent.
You know what today is, Dickson?
Yes, I do. It's Pearl Harbor Day.
That's right. What else is it?
It's TWiV 210.
Wow, you're good. You're really good.
I'm sharp today. I really feel
You're
on the
sharp.
top of my game.
Did you
here.
get a good night's sleep?
I did. I
Excellent.
did.
Also
Fine.
joining us, sorry, Dickson, were you going to say something?
No,
relevant?
I'm just going to say the cold that I had last week is still malingering, but it isn't keeping me up anymore.
You know, Harry Ginsberg used to have a saying about malingering. Do you remember what it was?
I don't.
Oh, something about the infection goes, but the malady lingers on. Do you remember that? I can't remember
He's
it.
right about it. Sure. He studied adeno-12 virus, right?
He studied many adenoviruses.
This is true.
Also joining us today in a triumphant return from North Central Florida, Rich Condit.
Hi, everybody.
How many TWiVs have you missed?
I have missed, I believe the count is 8 out of the last 9.
Wow.
I haven't been here since Chalberg.
People started asking where you were.
Well, I started asking the same thing. You know?
Welcome back.
Thank you very much. It's great to be back. It's wonderful. I've been looking forward to this. Sorry I didn't make it last week. Last week in particular, I wanted to be there, but, you know.
Well, last
stuff
week was
happens.
unplanned. You had a plumbing emergency. Yeah.
That's right. That's correct.
All right. Well, that's your yearly plumbing emergency. So.
In his house.
I hope.
Make it clear it's in his house, not
Yeah,
another-
not in his
in
body.
quotes,
Not in his
plumbing.
So
body.
everything's all good. It's all good. We're back.
Okay.
How's the weather down there? Good?
We got, boring. We've got 67 degrees Fahrenheit, 19.5 Celsius, and mostly overcast.
That's right.
Here
It's
really,
warm but boring.
It's really nasty here, right, Dickson?
Yeah, it's not nice out there. But it's
5 degrees?
It's almost the winter, So.
Cloudy? Raining?
Yeah, that's true. It is.
Oh, well. Also joining us today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody.
How are you doing?
Doing fine.
Don't have any plumbing issues, right?
No. And we have gray weather, 6 Celsius, 43 Fahrenheit.
Right.
How's the hanging wire in your front yard?
They came and
yes.
fixed it. I called the power company. They left a note and said it's AT&T. I called the phone company.
Of course.
The phone guy called me. The phone guy was very attentive, called me and said, I'm going to go out there. And then he called me when he was there, and he said, it's really the cable, the TV cable, and I'm just going to fix it because I don't want you to have to deal with somebody else. And then he called back and called my home phone. So anyway.
Wow.
Wow.
I'm impressed.
Amazing.
That doesn't happen out here.
No,
That guy, he would have left and said it's the cable company's problem.
Yeah.
it's true.
Also joining us today from Western Massachusetts, Alan Dove.
Good to be here.
You know, I'm getting my states confused. There's so many different states.
Yes. Yeah, so it's 38 Fahrenheit, 3 Celsius here, and yucky.
3, a little chillier than here.
Yeah, it's cloudy, it's drizzly, it's just gloomy.
It is quite. Pearl Harbor Day. What else is going on? Nothing really. What are you going to do? Cough?
Yes.
All right, have a good cough. I left them all in last week so people can see how much you cough.
I'll tell you what's going on. It's the holidays. I can feel this creeping up on us.
The holidays, yes.
Are you
Yeah.
going to see your offspring for the holidays, Rich?
No, they are kind of sort of coming here, some of them.
Okay.
It's their year to visit the in-laws, basically. So,
Right.
So if
but
you-
the in-laws are in Orlando, so we'll see them some.
If you need to get away, you can always do a podcast.
I'm planning on it. Yeah.
Now, we have a couple of follow-ups here from last time from you. Did you want to say anything
Sure,
about
we can
them?
do this quickly. It's actually from the time before, something like that. You had the episode where you discussed the notion of using silk to help preserve vaccines. And I have a particular interest in this because there was a postdoc in Joan Steitz's lab when I was there as a graduate student who worked on fibroin. And she actually raised silkworms in the lab.
Wow.
And I followed this with intense interest because she had to figure out how to do all this. The motivation here was that the worms, when they get into their fourth instar and are tooling up to spin their cocoons, go through a remarkable development to make this protein, fibroin. And they didn't even know at the time, we're talking mid-70s, they didn't even know what the molecular weight was. People tried to figure out the molecular weight by taking cocoons and trying to solubilize the protein, which is basically insoluble, and they came out with all sorts of little bits.
Right.
And Karen figured this out. I think I mentioned her name was Karen Sprague. She figured this out by dissecting out the glands, and this stuff is basically a gel in the glands. And she would dump it straight into guanidine hydrochloride and run these really low percentage gels and figured out that it was about 400,000 molecular weight. This stuff is like 90% glycine, serine, and alanine in a repeating structure that makes these fabulous beta sheets. And so the worms not only jack up the messenger RNA synthesis, which was of interest to make this stuff, but they jack up the tRNA synthesis to make enough tRNAs to plug in the amino acids for all of that stuff. And Karen was interested in the regulation for that. And the protein itself is really interesting. The wiki says that when it's exposed to air, it crystallizes and makes the fibers. And you can watch the worms do this. They spit out a little bit of this goo from their glands and pull their heads back, and you get this thread that then crystallizes in the air, and that's where the thread comes from. It's sort of crystallized as they spin the cocoon. And then, of course, to make the silk, you boil the cocoons and find an end and just unwind them. So the whole thing is just fascinating. I think,
right, because the whole cocoon is one fiber.
Yeah. Right.
Yeah.
It's amazing.
It is actually.
And Karen was doing stuff like she wanted to P32 label the messenger RNA, right? and that was tough. She tried injecting the worms, but she'd get it in the gut and then they'd leak because they didn't heal well. And she finally figured out how to feed them a diet. She had this artificial diet or semi-artificial diet that she made because they'll only eat mulberry leaves.
Right.
So if she had mulberry, she was the only person I knew who had a rototiller on her NIH grant because when she moved to Oregon, she had to farm her own mulberry trees, right? And but so she figured out how to mix P32 with her diet.
Sure.
And these worms were in the fume hood.
Wow.
Right, in a little plexiglass cage.
Wow.
Eating P32 labeled food. So she made hot worms, and it worked okay.
How very cool.
So my other follow-up was you discussed this idea of a vaccine that's missing the exonuclease in SARS-CoV. And what occurred to me, and I actually emailed Matt about this, I must have sent it to some wrong address or something because I would have expected he'd get back to me, but I haven't heard from him. What occurred to me is that this thing has the mutant that lacks the repair endonuclease or editing endonuclease must have a very high mutation rate. I would think it would be hard to grow that virus. And in particular, if I were in the FDA and evaluating potentially using this as a vaccine, I'm thinking, so you grow it up and what do you got? I mean, ordinarily you got a swarm, but here you got a super swarm.
Yeah, that's a good point.
And how do you know what one batch is or what another batch is? So I was interested in the ramifications of trying to, just the practical ramifications of trying to make a vaccine that has even a much higher than normal error rate. Okay.
Wow.
So
That's a good point.
Wow.
Yeah.
we can explore that down the road.
Yeah, traditional vaccine production would be an issue there.
Right.
So Matt's probably listening, so let us know, or someone else from the Barrick lab.
Right. By the way, did you know how the silk industry of the United States failed miserably?
Oh.
We tried to substitute the gypsy moth for the silkworm and introduced them into Paterson, New Jersey.
Oh.
And,
Oops.
yeah,
Oops.
they just sort of got loose.
Oh,
And here they are. Not
man,
like-
I was in Connecticut for one of those episodes.
Yeah.
I think we've talked about this before, where they just absolutely stripped the trees.
Correct.
There was one of those in Maryland when I was living down there.
Yeah.
Patterson, because it had the silk industry there.
Well, it had the right
Garment.
temperature,
Yeah.
and it had the right power supply because they were using Patterson Falls as an
I
energy
see.
source.
You know, I was born in Patterson,
I did not know that, Vince.
and I grew up there. I spent 8 years of my life there.
Wow.
Yeah,
Cool.
Patterson, New Jersey.
Wow.
Famous poet from Patterson, also, right?
Who are you going to say?
William Carlos Williams.
Actually, it was from Hawthorne.
Whatever, it's the same thing. Okay.
Lived in East Rutherford and taught at Fairleigh Dickinson, which is where I went to
He
school.
was a physician, and so my father knew
pediatrician, that's right.
his family.
He was a pediatrician.
And I have a book of his poems here,
Right.
which was my dad's.
Great.
Okay, we have two papers today, which actually have a common thread.
No pun intended, of course.
No, none whatsoever. Two common threads, actually.
All
One is they both solve a really old problem, right?
Right.
Right.
And the second is they use pretty cool biochemistry to do that,
Yeah.
right?
Yep.
Everybody
Yep. Yeah.
agrees?
And
Yeah,
they also
absolutely.
they involve particularly interesting aspects of virus using host proteins for other purposes.
Yep.
And finally, they're both about viruses.
Yes.
That's a given on TWiV.
Yes.
Right.
The first one is in eLife. This is our first eLife paper. eLife is a brand new journal. I bet you haven't heard of it, Dickson.
Well, I have now, but I didn't
eLife
up to 2
is
seconds
a
ago.
brand new journal supported by the Howard Hughes Medical Institute and the Wellcome Trust.
Nice. Those are two good
Let me
organizations.
read from their website. eLife is a researcher-driven initiative for the very best in science and science communication.
Right.
We promote rapid, fair, and constructive review. We will use digital media and open access to increase the influence of published works. We commit to serving authors and advancing careers in science. At eLife, publishing is just the beginning.
Ha.
This is a little while ago they had announced this, but they didn't have a name for it. They were wondering what they should call the thing, and I suggested they were starting a new open access journal because of their own, and I suggested it should be PLOS2 TOO. They didn't go for that though, so it's eLife.
No. They
So we just did an ad for them. Shouldn't they pay for it?
Yeah.
I mean, they're committed to science communication.
at least
right?
give us a little space in the journal for our ad.
It would be good. It would help us an awful lot.
Sure.
A little TWiV ad there in
It would be
eLife.
nice.
Anyway, these articles are all they're all Creative Commons licensed, so you can use the stuff as long as you give it attribution, original source and author. Source and author. So this is our first one.
Right.
There's a lot of controversy over this journal. I don't know if you guys have heard it, but
No, I have not heard.
people were complaining that it was just another elitist journal and would be hard to publish in and control people's careers. So,
But they have a statement in the beginning of their journal that says that's not what they're about.
Well, I certainly wouldn't say that the lab and the author. I certainly wouldn't say that the lab and the authorship of this first paper that we're looking at represents any kind of elitism.
No,
not at all. Not at all.
no, it's from the workers' paradise.
It's from,
Right.
From Beijing, China.
Ah.
Beijing University and a Chinese Academy of Medical Science, Beijing Union Medical College. Hey, why is it Peking sometimes and Beijing and other?
Peking is the older term for
Right.
Beijing.
So they don't change the name like Peking Union Medical College in Beijing. Why don't they change it to Beijing?
I think because they're stationary. They printed so many people
That's exactly what I was going to say.
that they couldn't possibly ditch all of that and make new stationery
Okay.
unless they just
Well,
crossed
and the
off.
other thing is their official name is not Peking or Beijing. It's the Chinese character for.
Can you say that, Alan?
No, I cannot. It's probably pronounced something like Beijing, but
Right.
Peking is the previous English
yeah.
Okay.
spelling of it.
And it's funded by the Ministry of Science and Technology of the People's Republic of China and also Science and Technology Bureau of the Beijing Municipal Government.
Excellent.
Some great science going on there.
Yep.
This is
sure.
great stuff. This is really amazing stuff.
This is a really good paper, too.
yeah.
Sodium taurocolate, co-transporting polypeptide, is a functional receptor for human hepatitis B and D virus.
Wow.
So people have been trying to get this receptor for a long time, and these guys cracked it.
Yep.
And
it has a number of authors.
Yes.
The first two are attributed as contributing equally, Yan and Zong, and the senior author is Wenhui Li.
Oh, you're not going to say all the ones in between?
No.
Yeah, okay.
Okay, I'll do
another
it.
one,
It's
two-
Yan,
okay.
Zhong, Zhu, He, Jing, Gao, Huang, Qi, Peng, Wang, Fu, Song, Chen, Gao, Ren, Sun, Kai, Feng, Sui, and Li.
Very good.
Cha-cha-cha.
Good job. Very nice. We've had a couple of episodes on Hep B, right?
Yep.
And
Yes.
I remember my former student, Michael Bouchard, did an episode with us.
And the person who discovered the protein from hepatitis B as Australia antigen is a graduate of our medical school, Baruch Blumberg.
Who got the Nobel Prize,
Who
right?
did?
He just died recently,
He
didn't he?
did. That's correct. That is correct.
The Australia that's a great story.
It is.
That is a really good story.
So that goes way back
Yeah.
into the 50s.
Right. Well, that's not why it's great.
No.
But it's really good. Anyway, big human pathogen, lots of people infected
Right.
globally, chronically infected, predisposing to liver cancer.
Yep.
There is a vaccine, of course.
Yes.
But one of the big problems, so this is a virus with an odd genome, double-stranded DNA with a gap and a piece of RNA and a protein linked to it. Has reverse transcriptase as part of its life cycle. Very cool.
One of these 7 methods for viral replication
yes.
that David
You
Baltimore.
still remember, Dickson?
Of course I do.
Someday we'll get back to Virology 101.
That'll be good.
I was thinking about it in the car this morning, and then I had an accident. Almost had an accident.
Really?
Just kidding. I shouldn't joke about that.
No.
And it's an enveloped virus with glycoprotein, this S protein that is responsible for attachment to cells. And it has a satellite associated with a delta, hepatitis delta virus, which uses the glycoprotein of Hep B. So presumably the receptor would be the same. And that's what we'll see from this.
Delta is a small circular RNA element that encodes, I guess, one protein that's required for its replication, delta antigen, but beyond that, steals this surface protein for its encapsulation from Hep B, and so is entirely dependent on Hep B for propagation and exacerbates the hepatitis B disease significantly.
Hey, Rich, is hepatitis delta living?
We're going there again.
I just want you to tell me yes or no.
It is when it's in a Hep B infected cell.
Okay, fine. But this little piece of RNA, it can't be alive. Come on.
I know. I'm getting so confused by all this.
It's okay. I couldn't resist. I'm really sorry. Put you on the spot. Hepatitis delta.
That's what you get for staying away so long.
I mean, actually, it's amazing. The whole thing is amazing. These little pieces of nucleic acid, I don't care what virus it is.
Right.
It's just a little piece of nucleic acid causing all of this turmoil.
You sure?
God.
This receptor, Dickson, was identified by zero distance cross-linking.
Really?
You know what that is?
Well, no. I don't have a clue.
So-
even from the title, I couldn't tell you.
There's a protein that attaches to the host cell in the viral
Zero
glycoprotein.
distance cross-linking?
So they have a little peptide from it that's already been known to compete with virus attachment. So they figure this thing is attaching to the receptor. So they put cross-linkable chemical groups onto this peptide
Right.
and bind it to cells. They also put a tag on it. They make antibodies to it. And then they add it to cells. The cells are important. We'll talk about that. And then they put UV light on it, and it cross-links to something that's of zero distance from this peptide.
So it has to come in contact with the thing that it's going to cross-link
Right.
to.
Got it.
I think you can make cross-linkers that will cross-link at various distances from the protein or whatever it's binding to. You can buy them at various distances, but this is zero, so this is pretty cool.
Right. This is a photoleucine, so it has to be snuggled right up against whatever it's going to cross-link to.
Wow.
And then
the
you hit it with
peptide-
UV light and it cross-links.
Yeah.
the peptide that they're using is like 40 amino acids from the N terminus of the S protein. It's meristylated, so it's got a fatty acid on the N terminus, and the tag on the C terminus is a biotin. And they've made this synthetically with these two residues that have the photocross-linkable leucine in them. It's incredible.
It's beautiful. Both, and this peptide inhibits both delta and hep B.
Right.
And in fact, they mentioned somewhere that delta has always been a surrogate for hep B binding. Now, I have the suspicion that it's probably easier to get delta than hep B particles.
I think they can make delta they make it with a couple of plasmids, one that makes S and one that makes delta.
So could you make a dysfunctional delta that's missing a few amino acids and interfere with the attachment process of both of them then?
Sure.
Okay.
Yeah, I'm sure you could.
Just a random thought.
Dysfunctional delta. I like that title. But that's not what this is about, though.
No.
I think I flew on that airline.
That's right. I used to live in it.
So this is such a beautiful approach. I'm sure it took a long time for them to work it out. But they cross-linked this peptide, and then they pulled the peptide down with antibodies to it that they have made.
Right.
And then
Actually, streptavidin to the
biotin, sorry. What were the antibodies for? Just for detecting?
For detection as well.
Yeah.
Well, they actually at some point I think have a double purification that uses both the antibody and the biotin label, like two different tags. So they use it in a variety of ways.
So the peptide was biotinylated, and then they used streptavidin, which is bound to beads, which binds biotin really high affinity and pull down whatever's attached to the cross-link to the peptide. And they basically get the viral receptor this way.
Cool.
Get the protein. Right. The cool part is they get a protein, they do mass spec to sequence it, and then at the same time, all right, so these cells that they're using are hepatocytes from Tupaya, tree shrews.
Yeah, if we thought that they were doing a lot of work already, it gets better.
They have a colony of tree shrews.
They established a colony of tree shrews to work on this
So
thing.
they say that 10 times fast.
No thanks.
Tree shrew. And then they use the liver cells, make primary hepatocytes. They add the peptide cross-link and get the receptor out of them. But, there's no genome of tree shrews, so they sequenced all the mRNAs of this. The transcriptome they did at the same time. This is just they did everything.
One of the big problems has been that the host specificity of HBV and HDV is very stringent, very narrow. It infects humans and chimpanzees, and this one other creature, which is this tree shrew. And of course, getting primary hepatocytes from either humans or chimpanzees is almost prohibitive. So in order to do a study like this, you need a lot of cells that you can use. So they established this colony of tree shrews. Amazing.
And it can't just be primary hepatocytes. You can't just pull them out and use them a few days later, apparently. You've got to have fresh primary hepatocytes in order to be able to infect it with hepatitis.
Right.
So then they did the transcriptome of these hepatocytes so that they could say, what if we cloned out here?
Right.
And they pulled out this sodium taurocholate cotransporter molecule,
which is apparently a fairly important part of bile metabolism.
Right.
And cholesterol metabolism too.
Yeah. Which of course, it's in the liver, so
yeah.
And it's
highly liver specific, so that
yes.
winds up making sense.
Makes sense for hep B. So what do they do with this? They show that they clone the whole gene for this NTCP, express it, show that it binds SS, and that if you express it, if you knock it down with siRNA, you abrogate binding of hep B and hep delta to cells. So do that by RNA silencing. And the cool part, what I think, is they express it in cells that normally are not infected by HDV and HBV. So I think this cell line, HUH7, which we heard about today, Dickson.
We did.
In context of hep C.
This is true.
And hep G2, these are both hepatocyte cell lines.
Right.
But they're not infected with hep B. But if you put this NTCP in it, boom.
How
get
about that?
infected.
How about that?
And this is going to revolutionize studying this because this virus has been really hard to work with because you
Yep.
can't grow it.
So not only did they find a receptor, they now have a cell culture system for Hep B.
Yep.
Right.
And maybe if they put it into mice, they'll have an animal model.
Maybe.
I know there are like woodchuck hepatitis B viruses, and they can use that. Some people do ducks.
But isn't
ground squirrels.
there a humanized mouse that has a human liver?
Yeah, I'm sure there is.
No, we heard about that 2 times ago.
Yeah.
There
So-
are humanized mice, but they are really, really expensive.
Aha.
So we heard about one in our journal club this morning where basically you implant a tissue engineered thing that's from some human tissue into the mouse.
Okay.
So if you imagine you have to do that surgery for every mouse.
Oh, well.
Yeah, it's not easy.
So now you can take a liver cell line, make it infectable with Hep B.
Nice.
So I was wondering, there are these other model systems. There's duck hepatitis
Right.
virus and
Right.
woodchuck hepatitis virus.
Ground
I'm
squirrel,
wondering
yeah.
why people have not been successful in doing this same thing with those, because there you do have more access to appropriate material.
Yeah.
Actually, I wonder if the receptor's even the same in those. I suppose we'll find out pretty soon.
I'm sure they're all looking at it right away. Yeah.
Right.
Interesting.
I think one cool experiment they did here was to show if you swap. So they have this crab-eating monkey. I love that name, crab-eating monkey, Macaca fascicularis, which doesn't bind HBV. But if you put in, if you take the NTCP from that species and just substitute some amino acids from human, you can make it bind Hep B
In the cells.
in the cells, not in the crab-eating macaques.
Not in the crab-eating macaques.
I guess they eat crabs, right?
Yeah. Yeah, that wouldn't be such a great model system
No.
because it wouldn't be any better than raising tree shrews, probably.
Now, this isn't the whole thing, the whole story, because even though you can infect these various cells now, they don't make a lot of virus. I mean, even the primary hepatocytes don't make a lot of virus.
Yeah.
So there's still post-entry steps that are not very efficient, and they have to figure out what's going on there. It's the same with Hep C in mice. They can make Hep C enter mice by putting all these human receptor molecules into them, but the virus doesn't replicate very well still. So there's still post-entry things, although I don't understand why in a primary hepatocyte that would be the case, right? But I guess when you culture it, things change, right?
Yeah, that's, I think, one of the most interesting experiments here is this. They did this experiment where they took primary hepatocytes and, as Alan suggested, put them in culture and then watched the expression of this receptor over time. And it decays very rapidly,
Right.
which provides a good explanation as to why people have had trouble with this before. And God knows what else changes. A lot of stuff.
Yes,
During
absolutely.
the first few days. And so there's still a lot of work to do.
But now they can start addressing some of those.
Right.
If you're looking for the internal block, it's much better to do that in a cell culture model.
Right. Absolutely.
So this NTCP is mainly expressed in the liver, and that, of course, is where hepatitis B virus replicates. So I guess we can say the receptor is controlling the tropism of that virus, right?
Would make sense.
Absolutely.
Well, yeah, it's probably a major part of the tropism.
Yeah, maybe there's something else.
Malaria, by the way, even though that's not a virus, has a stage that only infects liver. It's called the sporozoite. And the receptor molecule is on the endothelial cells of the sinusoids of the liver. And so that's the reason why it can circulate throughout the entire body, but it only goes to the liver because that's the only place it sticks. So that's the same here, too. There are some parallels in biology.
Are those living, Dickson, those malarias?
They are. They have a metabolism, Vince.
That's one of your criteria, right?
I would firmly stand by that one as the number one criteria. That's right.
I think I listened to it again, and somebody said that, once it's outside the cell, if there's nothing enzymatic going on, then it can't be alive. But there are some examples of viruses after they're outside the cell having some enzymatic things going on.
Yeah.
So
That's true. That's a good one. Yeah. There's always an exception.
But that's a crossover. That's halfway.
gray, we call it gray.
That's right.
It's a half-life.
It's a half-life. There you go, Alan.
Yeah, so
Dynamite.
this is a great paper, I think.
This
cool.
this has been a people need to appreciate that this has been a
Summary.
decades old
Yeah.
challenge
Yeah.
that people have been trying to. A lot of people have crashed and burned trying to isolate this receptor, and it's been a very, very tough. So this is a real triumph.
And I must say, this eLife paper has a lot of experimental detail. I don't think there's a page length limit. I might be wrong, but I don't think if this were a Nature paper that it would be as detailed as this.
No, one of the things they say in their little rap about it is that the format for the publishing online, since it's entirely, and there's no print, it's entirely online, they aren't really restricted to space or number of figures. They're committed to giving all the detail.
Yeah, this is a 28-page paper.
Wow.
Yeah, there's no such thing
Great.
as supplementary data.
Great. That is good. I really like that.
And is there no page charges then?
I don't know about that.
Just
I don't know how it's funded.
maybe it's a time thing.
I'll
However long
bet, I'll bet there's a page charge. Yeah.
Yeah. So let me ask a naive question here, which has some practical application. Now that you know what the receptor is for hepatitis B and D viruses, does this suggest an interference strategy for clinical?
Sure.
Theories
Doesn't
and
suggest
theory.
it'll work.
Yeah, but you got to watch out for the side effects. Because remember, this transporter is important, right?
Well, yeah, but what if you flooded the animal with a soluble receptor, peptide?
Yeah, you could. Well,
My
peptides
guess,
Go ahead.
my guess is that sort of approach, that people are already looking at that, because it's been known
Yeah,
for some time
yeah.
that this N-terminal peptide will block
I see. I see.
entry, even and that was fundamental in getting this
I see.
study going. Even if they didn't know what the receptor was, they did know that this peptide would block it. It's nice now that they've got a mechanism
Got it.
that helps them pursue it further. But my guess is there are a lot of people exploring that sort of thing.
Okay.
Yeah, the thing that you might try with this result is to then take purified receptor peptide and flood the patient with that to see if you can saturate the virus-binding
that's
sites. But
just what I said.
yeah,
I thought.
There
Maybe I
is,
didn't say that.
okay, we understood it the other way around.
No, I meant to say that.
There's also a nice commentary in the eLife by Chen and Yi. So that we'll link to that. That's really very nice. And the cool thing about this is all the pictures we could use on our website if we wanted to, as long as we attribute them. So thank you, eLife, for that. That's our first one. We'll see how many more we do.
Yeah.
The next paper is something close to home. Poliovirus. It's a paper from my old friend, and yours too, Kathy, Bert Sembler.
Yes.
Maybe everybody's friend.
Yeah.
Bert's my friend.
Okay, you could be everybody's friend.
As a matter of fact, Bert was in Eki Wimmer's lab, I think, still, while I was there with Niseen Musashi doing a postdoc. I think he was just kind of out the door leaving while we were there, something like that.
Was Stan Fields there when you were there?
Yes, I believe so.
I just met him this week. Stan Fields, of course, invented the yeast 2 hybrid assay. So this paper, Bert Sembler is a professor at UC Irvine, works on polio, rhino, and picornas in general. And the name of the paper, published in PNAS, is an RNA virus hijacks an incognito function of a DNA repair enzyme.
And the authors are Virgin Slane, Janet, so that's Richard Virgin Slane, Janet Razovich, Carrie Fitzgerald, who I know, she used to be at University of Michigan, Tuan Ngo, Wayne Chow, Gerbrand van der Heijden van Noort, Dmitry Filipov, Paul Gershon, and Bert.
This has to do with, this is such a nice old story. So when I was a postdoc in the Baltimore lab where the finding that this is based on was made. And so let's just review a little. Poliovirus, of course, and all the picornas have a plus-stranded RNA genome in the capsid, and it's linked at the 5' end to a little protein called VPG. And this link is via a U residue in the RNA to a tyrosine in the VPG protein. Now, way back in the late 70s, early 80s, a graduate student of David's by the name of Victor Ambrose found that in cells there is an enzymatic activity that will cleave VPG from the RNA. So it looked like as the RNA is put in the cell by the virus during infection, shortly thereafter, this protein was cleaved. And Ambrose purified this protein, showed that it cleaves the linkage, but he could never identify it. Ambrose Patterson in Baltimore, 1978. It was a Cell paper, actually. Remember that. So for years and years, no one could identify this protein. I know people have tried, and some people would purify it, and they found that the N-terminus was blocked, so they couldn't figure out the sequence, and that's the last I heard of it. But obviously, it would be interesting to know what it is, because then you could ask, is this really necessary for viral replication to take this protein off, right?
Mm-hmm.
So what is VPG there for to start with?
Well, VPG acts as a primer for RNA synthesis.
Ah, right.
So the polymerase puts 2 U residues onto it, and then it primes RNA synthesis at the poly A tail.
Okay,
Right.
so it's kind of like the adenovirus terminal protein.
Yeah.
Mm-hmm.
And so all the newly synthesized RNAs have this on it. But when the RNA first comes into a cell, it's cleaved off. Now, nobody understood why, because you can make... So if you take cloned polio DNA and you make an RNA transcript, it doesn't have VPG on it, but it's fully infectious, right? So nobody understands why VPG is there or has to be cleaved off. So this is a big step in this direction. So what Bert did was to go back to this problem and try purifying it. And basically he developed an assay where he had labeled VPG on viral RNA. It's labeled with S35 methionine. So
And they made that like by infecting cells.
Yeah.
It's metabolic labeling, right?
old school biochemistry.
Old school
Wow.
stuff. Right?
Wow.
That's right.
And that was
Infect cells
key.
in the
That was
Yeah,
what?
absolutely.
That was key to the success. In fact, there's a whole previous paper to this in PLOS One that's open access, and they talk about a lot of the previous studies. People had taken that same molecule and then cleaved it and used that as their substrate to try and purify the unlinkase. But in fact, it was much better to use the full-length molecule, and that
Right.
was probably a large part of the success in this biochemical approach.
So I basically fractionated cells and eventually got a pure protein. You know, they went through all kinds of columns, which I don't know if many people do anymore.
Oh man, this just really blew my mind. I sat down and Susan and I went through this in excruciating detail because we've been through this. And to watch what they did is they didn't start with much stuff. 5 mils of packed HeLa cells. Of course, they've done the prep. You know, they've been doing this for years. They've done the prep obviously many times, and the representative prep is in the paper. But the 5 mils of packed HeLa cells is not a whole lot. They got 200 milligrams of protein that they start with, and then go through 5 or 6 different columns and wind up with a microgram. The other end, amazing. But of course, nowadays, it used to be that you had to purify up 50 milligrams or something like that to do anything with it. Now,
No, you can
all
do
you
it
need
there,
to do is identify it, and then you can clone it and express it, and you're okay.
Right. So I, too, enjoyed seeing this purification table because I remember seeing those all the time through my graduate postdoctoral years, and you don't see that much. And the other thing is that a PNAS paper always used to have the enzyme commission number for an enzyme, and it wasn't there. And so I looked up what it is for this enzyme, and it's 3.1.4.1.
Excellent.
Which, yes, I emailed Bert and said, yeah, the first digits of pi, that's pretty cool.
Nice.
That's cool. And I want to elaborate on the assay a little bit, too, because it's really cool. So you have the S35 labeled protein hooked to the RNA. And if you run that on a gel, on a fairly high percentage gel, these are 15% trypsin gels, it's really all at the top. And you make an autoradiogram, right? So you're seeing the labeled material, and it's all stuck at the top. And if you treat it with this enzyme, and they have to treat it very quickly, it's like a 30-second reaction. it will cleave the protein from the RNA, and the protein then runs down on the bottom of the gel. And the reason you have to be careful with this is that, in particular with a crude extract, it's going to have a bunch of ribonucleases in it. And if you treat it for too long, it's just going to degrade the RNA, and the protein will run down towards the front of the gel. So it's going to look like it's the same activity. But they can actually discriminate between ribonuclease and the real activity by two criteria. First of all, in these high percentage gels, if you treat with if you RNase the substrate, you wind up with a protein that still has the U attached, and that runs just a tad slower than the thing that's authentically cleaved. Secondly, you can do a northern blot with the gel in the end and show that the RNA is still intact on the top of the gel after it's been cleaved with the enzyme. So I thought the assay was just fantastic. It's cool.
Yeah.
So you only need a microgram or less because they do mass spec nowadays. In the old days, when you had to do, trypsin fragments and Edmond degradation, you just needed many, many more.
Now you just squirt it in the magic box and get your answer.
So they did that. They got a little bit of the protein mass spec, and they identify it as 5' tyrosyl DNA phosphodiesterase. It's a DNA repair enzyme. And there are two versions of this. There's a 5' tyrosyl and a 3' tyrosyl version, two enzymes in the cell. And my understanding, now you're the topo guy, Rich, my understanding is when topoisomerase unwinds double-stranded DNA, sometimes the protein can get attached covalently to the DNA.
The DNA enzyme covalent attachment is an obligatory intermediate in the reaction.
Okay.
Okay, so there's an obligatory intermediate where there's a phosphodiester bond between one end, depending on the enzyme, one end or the other of the DNA, and a tyrosine in the enzyme. And in topoisomerase nicks DNA, and then the DNA unwinds, and then it reseals it. And so the enzyme stays there in this transient intermediate, and then when it reseals it, it just reverses the original reaction.
So when it's nicked, then it goes on to topo and then unwinds and then back again.
Bingo.
Okay. And these enzymes mediate the cleavage of the protein DNA bond, right?
Yes. Yeah. Apparently what can happen is that for various and sundry reasons, the enzyme may not go to completion, the reaction may not go to completion or get stalled or something like that. Then you got a problem because you not only have a nick, but you got this big old protein sticking off it, and you need to repair that. Ultimately, you would get a double-strand break if that's not repaired. So these enzymes are there to look around and find any of these things that have stalled or screwed up and cleave the enzyme off so that now you only have a nick and you can complete the repair. So they are genuinely repair enzymes to repair issues that arise from topoisomerases or topoisomerase mechanistically like enzymes that don't finish the job.
Right. So that enzyme, the 5' version, is what's cleaving the VPG RNA bond. So apparently this DNA enzyme can also hit RNA, which is pretty cool. Yep. And it's not known that it does this in the cell, but it could be. I mean, they suggest that this is an old enzyme from the RNA world, and maybe it still has some RNA cleavage activity. So that's pretty cool. So they express the protein from a clone, and they show it cleaves the VPG RNA for polio and rhinovirus, right? So that's cool. They didn't do the knockdown. That's, I suppose, the next 5 papers.
Yep.
But I would have loved to see knocking this down and what happens. Is it needed for replication or not?
Yeah.
Because that's really the question.
What's your bet? Come on, let's put bets on this.
Oh, I think it's needed for replication.
Yeah, gotta be.
They'll see an effect on replication, and then they have to figure out why, right? At what level. Now, what I think is the really interesting question here, which they start to explore, right? So in the beginning of infection, VPG is cleaved off, but at the end of infection, you package RNAs with VPG on them. So if this enzyme is in the cell, how do you deal with that? And they do some confocal, and they say that later in infection, the enzyme, this DNA repair enzyme, is not at the same place as where viruses are replicating.
Right. So this is really nice fluorescence microscopy, where they're showing they can identify where the protein is in the cell and where the replication is happening. And they show that, like you said, they're in two different places.
But I can't tell whether the mock-infected cell is screaming or laughing.
Yeah, it's a good one, isn't it?
Yeah, it is.
Yeah. It's a Munch. Is that his name? The painter.
Yeah.
Right, the scream.
Edvard Munch. Yeah.
This is figure 4A for those of you who are going to look at that.
Yes.
So their idea is that there's a compartmentalization. All right, but I have another idea. What if the damn thing is cleaved by polio late in infection or inactivated in some way? That would work too, right?
That would work too.
Yeah.
I know compartmentalization, I suppose, would work, but also modification by the virus, that would kind of be neat. And I don't think they mentioned that, but maybe a cleavage or modification of some sort would be. So Bert and guys, well, Bert doesn't listen to this, but
he will.
Anybody in his lab, he will.
He will. But in the previous paper, the one in PLOS One, they did show that the virus infection does not change the activity of the enzyme throughout the course of infection.
So if you purify it earlier and later, there's no difference.
Right.
Yeah, but who knows? Maybe.
So when is it added onto the virus just before it leaves the cell?
During RNA synthesis.
Okay.
So whenever an RNA strand is made, the VPG is put on right there.
All right.
I mean, so that happens, within an hour, I would guess, of infection. So you've got to find. So compartmentalization probably makes sense.
Polio does establish compartments for replication, does it not? Membranes?
Yeah, it's on the surface of membranous vesicles, right?
Right.
So that could be one way of doing it, yeah.
Yeah, all it would have to do is exclude this enzyme from those complexes
Yeah.
long enough for the genomes to get packaged.
Right. So I guess, Kathy, the one thing you could say is if they purify it early and late or just use a crude extract of cells early and late? Because if they purify it, you could argue if there's a inhibitor, it's leaving it or something like that. So who knows?
In that previous paper, if that's what you're talking about.
Yeah.
So it's a protein chromatography fraction enriched for the unlinkase activity.
Yeah.
For various times with. Anyway.
So how would you prove that the compartmentalization is what is regulating the activity? Is that difficult, right?
Yeah.
Maybe you could make a altered protein that doesn't get compartmentalized and show that it cleaves all the VPGs off or something.
Right.
Plenty.
Or an altered virus that doesn't compartmentalize it.
Yeah.
That's going to be tough, though.
It is tough, but plenty to do there. Lots to do.
Mm-hmm.
The thing is, now that they've published it, everybody can do this.
Right.
Yes.
Right.
So, but that's fine. That's how science should work, right?
And as you said, Vincent, it was a long-standing question.
Yeah.
Activity's been known for a long time. People have worked on it, and so.
And
nice.
Bert just, thought we should do this, which is good because maybe no one else would have. Right?
Mm-hmm.
And then we'd never know. So
it's a fairly high-risk project to take on.
It is.
Very, very high risk that this wasn't going to work.
Yeah. So that's another cool paper. 2 old questions answered.
Yes.
Yeah.
By classical biochemistry techniques.
Really nice. All right, let's do a few emails. Would you like to take the first one there, Rich, since you haven't been around for a while?
Okay. Jamie writes, "Greetings, Vincent and TWiV adventurers. I am a 28-year-old medical biller who has sort of floated through life not really knowing where my passions lay. Join the club. I'm not even sure how I came along TWiV, TWIP, and TWIM. To be honest, all I know is I somehow listened to one episode of TWIM, and through TWIM, I found TWiV, and I was hooked. There is quite a bit of the program that goes over my head, but I can normally get the gist of the conversation. Today, I emailed the state university I lived near to talk about enrollment.
Great.
I already have some credit in healthcare administration, but now I'm thinking of diverting those, if I can, towards public health. I'm not sure where I want to focus my studies, but I decided if I only get to understand what you lot are discussing, I've gained a lot. Maybe next time I write, I'll be able to tell you where in this large field I'm leaning. Again, from someone who is just beginning this path, I love the show and keep it up. Isn't that nice?
It is.
Cool.
Very good.
I also noticed that in his letter he says, but I can normally get the jest of the conversation, not the gist. It's spelled J-E-S-T, so I think, Rich, you ought to reread that.
Okay.
Maybe he's hinting at the fact that we use a lot of humor in our educational process.
Right.
I'm not getting these notes here.
Okay, so I think I was thinking getting the gist of something might be an eggcorn.
Oh,
What's an eggcorn?
An eggcorn is a common misinterpretation of a phrase or a word.
Ah,
So eggcorn itself is one. Eggcorn for acorn.
indeed.
Getting the gist of something would be another.
Arg.
But we're not, Jamie. We're not picking on you.
No, of course not.
And very, very cool that you're finding this interest. So
yeah, it's great.
Yeah.
Kathy?
Okay, this next one is from Cristiano. And I think he wrote to us, I know he wrote to us before, and we have read that email on the air before. So
Right.
he was a little worried that maybe we hadn't or weren't going to do that.
Right.
Anyway, he's at the University of Otago. He's a postdoc, and that's in Dunedin, way down the bottom of the South Island of New Zealand. Thus, you can assume TWiV is everywhere, including the end of the world. I work at Andy Mercer's lab, who, by the way, asks me to send his regards to Rich Condit.
Back at you.
Besides being a postdoc, I'm also an aspiring science communicator specializing in natural history documentaries. I've suggested the topic of viral infection in the event of xenotransplantation before, and he thinks with no success. But in fact, we did
talk about it,
just
right?
talk about that on TWiV 201. It just
this is this fun documentary about the pigs and the xenotransplantation.
yeah, and then the mayor from the town.
Yes.
Yeah,
Yes.
so yeah.
We had all these pigs. Yeah,
That's
he's
right.
great.
Yeah, so these are the same links he sent before, right?
Right.
Okay.
And then he says, I would also like to share another one of my clips. While listening to one of the TWiV programs, I was inspired to work on an animation to explain the difference between prebiotics and probiotics. This was my first attempt in the world of animation. And he gives a link to a YouTube. And he says, I hope you consider my request. Love both shows. Best regards from the end of the world, Chris. P.S. Congratulations on 200 TWiVs. So this YouTube is well worth looking at. Those of you that haven't done that yet can't click on it right now. It'll just start popping up live and you'll laugh too loudly. It's really cute. It does explain very nicely the difference between prebiotics and probiotics. And the microbiome is an intense area of research in my department. So I had any number of people I could ask. And I asked one and said, is everything in here scientifically accurate? And she said, yes. Even the line where he says, bad bacteria cause diseases, even cancer. Because if you include H. pylori as a bad bacteria,
Sure.
then that's true.
Absolutely.
She was a little concerned because the link between the microbiome or the microbiota and cancer, causality hasn't been established. But she said that this is really great. She wants to use it in her classes as well.
Cool.
So
Great.
check it out.
Cool.
Just know that one of your TWiVers is going to be in your area in about a month from now, Chris. So my wife and I are going down to the South Island for a vacation starting on the 18th of this month. And we look forward to it.
Sounds great.
Yeah.
So H. pylori is probably early in life a good one, and then it becomes bad later in life, right?
Yeah.
That's our current thinking.
Is that it?
Okay.
Yeah, because, this guy Marty Blazer here in New York thinks that early in life it prevents asthma. And he has good data from a mouse model. There's good epi data in people. But then in your 50s, it can cause stomach cancer. So go figure. Alan.
Okay, I'll take this next one. Ken writes, I'm a computer scientist who, as part of an open educational resources project, implemented a web-based simulation of the Spanish flu pandemic. And 6 months ago, while doing background research, I stumbled upon TWiV 15 and have become a regular listener ever since. And he gives a link to this this model. So it's a computer model of the epidemic, and you can change various parameters of the 1918 flu. You can say, well, what if World War I had gone on longer? or change
No,
different characteristics of it and see what happens. And then he continues, I do have a question about Professor John Oxford's theory of the origin of the Spanish flu in a British army camp in France, and he provides a link to the paper. The virus would need to have infected people at the camp for over a year without significantly spreading elsewhere. For the computer model, this meant a very narrow range of possible values for the transmission odds, since a little lower and the infection dies out, and a little higher and it spreads widely a year too early. Professor Oxford explains this by the heavy flow of new recruits into the camp, so there's always a good supply of susceptible hosts for the virus. Outside the camp, it doesn't spread well. My question is whether there are known cases where a small fraction of a population were infected with a short-lived virus, such as the influenza virus, for a long period without the infection spreading or dying out.
A year without spreading, or I don't know how that. I don't think that could work.
Seems
unlikely.
Pretty unlikely.
Right. That's his point.
I looked at this paper, but it doesn't seem that it would work because it was simmering there for a year. And if it was really the 1918 virus, which was pretty bad, I don't know why it would be.
And people coming in and out of the camp?
No, they would have carried it off with them. And
I would think so. Yeah, so I think it's unlikely. But who knows? Who's going to know?
Who's going to know?
Okay. Dickson.
Here we go. Fernando writes, Hi, TWiversarians. That's the first use of that word. Just got back from 2 weeks of ski mountaineering in Argentinian Patagonia and have listening to TWiV 199. In Argentina, some mountain huts, refugios, were closed because of hantavirus concern from the mouse overpopulation that was caused by the most recent instance of the coordinated bamboo flowering discussed in this paper. And he lists a paper. There are quite a few other topics there are quite a few others on the topic. As for Yosemite, answering some questions you asked in the podcast, last winter was exceptionally dry in the High Sierra. Central Station plot and then he gives a website address. All but one of the cases involved people who stayed at the signature tent cabins in Curry Village, which are double-walled. The tent cabins were where the victims stayed were signature model built in 2009. Unlike the other 3,000-plus tent cabins at Curry Village, the signatures have an insulating wall of drywall in addition to canvas siding. The two have a gap between them large enough for hantavirus-carrying deer mice to build their nests, park officials said. So he leaves us with that query as to whether or not the actual construction of the huts could have favored the outbreak of Hanta in both of these two places.
Apparently it
did. Apparently so.
And the take-home lesson is get the cheap tent.
Sleep outside under the stars. And
although one of the cases, as I recall, was a hiker who didn't stay in the signature cabins but went into the back country and stayed in a lean-to site that they had there that also happened to be infested with mice.
That's very funny.
This paper he sent, Myths and Facts on Ratadas, Bamboo Blooms, Rainfall Peaks, and Rodent Outbreaks in South America. Ratadas are rodent eruptions that have been recorded since the 16th century, and these correspond with bamboo flowering and seeding, so that makes the mouse population grow.
That's the ecology
right?
of these diseases.
Rainfall,
right.
Bamboo, mice.
Yep.
It's very cool.
Cool stuff.
So they basically went out and confirmed that this happens.
Sure.
It's pretty cool.
Yes.
I would never have seen that paper. So Thank you. Okay, where am I? I missed. And it's up to me now.
Sarah. Next one.
Sarah writes, I came across your site as I was searching sites related to an article I wrote called 10 iPhone Apps for Tracking Kids Vaccination. Thought I would reach back out to you to let you know about the article and see if you would be willing to share it with your site's readers. Now, I get an email like this a day of all sorts of things. People just want clicks, but this one is actually looks good.
Yeah.
I mean, it's a blog called Nanny Pro, but 10 iPhone apps for tracking kids' vaccinations. I didn't use any of them, but because my kids are all past that, but maybe some of them are good.
Right.
All right, so figured we'd pass it on. Maybe it's just, link bait, but who knows?
Now, there was, I saw, I forget where, an article recently highlighting how confusing it is for parents to try and understand the vaccination schedules from CDC, and it really adds to all of the turmoil over getting kids vaccinated. So a good app that sort of explained this and helped people track vaccinations. I haven't looked at any of these, so I don't really know, but might be of real value for parents to understand what's going on.
As a parent who's dealing with this recently and currently, I come at it from a different background, obviously, but even for me, it's tough to keep track because there are so many of these vaccines, and they have different requirements and schedules. And gee, is my kid up to date on this, that, or the other? Now, we have a good pediatrician, and obviously I know where to find this information if I forget it. But it is very, very true that the vaccination schedule is opaque to most parents.
Yeah.
As long as these apps are accurate, right?
Yes, as long as they're accurate.
So we don't know.
Yeah.
So
Well, one of them is a CDC vaccine schedule. One's a Johns Hopkins app, number 10. So those, seem to be like they're going to be
go with a
reliable.
credible one, and you'd probably be okay.
Yeah.
Yeah, those are the ones that should be free, right? The ones from CDC or...
You bet.
Yeah, the Johns Hopkins ones, number 10 is free.
Some of them aren't, so I would go with those first. CDC vaccine schedule, yeah. All right. We are back to Rich.
Yeah, but Rich isn't back. Rich is playing with his iPhone. So here we go. I have a vaccine app that I use all the time. That's really pretty good. Anyway,
So you could keep track of immunizations? Is that what it's for?
these questions come up as in courses or talking to people is what's the vaccine schedule for such and such. Okay,
Yeah.
So it's there.
Okay.
Stephen writes, in TWiV 200, you mentioned alternative careers for scientists. There are two possible misapprehensions about science writing that might occur to listeners. One, to be able to switch from science to writing about science, you must be a natural writer. Two, anyone can write. Both are wrong. You can learn to write, and you need to learn to write.
Hear, hear.
There are several science writing and science journalism programs in the U.S. I'll suggest one as a listener pick of the week, the one I completed many years ago, the UC Santa Cruz Science Communication Program, and he gives a link. Here's a quote about. Here's a quote from the About page. The science writing program at UC Santa Cruz has produced professional science writers since 1981. The program is one academic year long with internships throughout the school year and the following summer. It focuses entirely on practical training through classroom work and diverse internships, and it's the only graduate science writing program in the nation that requires a degree in science and experience in research. The site includes pages about alumni and their writing.
And I will add to this, NYU has a, I will say, outstanding program in their journalism school, which I used to be an adjunct professor in, but it's not anymore. It's the Science, Health, and Environmental Reporting Program, SHRP.
Cool.
And MIT also has a program very similar to that, which I see, Kathy, you've provided the link to as well. So I think we ought to probably put... couple of those links in there.
I think Seth Mnookin teaches in the MIT.
Yes,
program,
I think
right?
Seth is at MIT.
Yeah.
And I have firsthand experience with SHRP, and I've heard very good things about the Santa Cruz program as well. So those are all good. And Kathy, yeah, you mentioned Johns Hopkins. That's another one I've heard of.
Mm-hmm.
Yep, here he is, Seth Mnookin.
Columbia's journalism school has a good one as well.
And I taught in your course, Alan.
You did?
Gave a
Yes.
lecture on polio.
Yes.
The woman at Columbia's name is Margarita Holloway. She used to be associate editor for Scientific American.
How did you learn to write, Dickson?
By the seat of my pants.
Good.
No, actually, I had some wonderful English teachers in undergraduate school that really cared about writing and actually read what you wrote and made the the right corrections whenever you did something wrong, and eventually you have to write every day. It's like it's practice. You have to just write, Alan. You have to just keep doing it. You can't let up. You have to keep doing it.
You know, I had great training in K through 12.
Is that right?
People actually taught grammar.
How about that?
So I diagrammed sentences,
Yeah.
and I learned to outline.
Right.
And all of that stuff applies today. I understand the
Right.
parts of speech and how they relate to each other.
Yep.
And I was hearing Alan talk about this last time. I use outlining. It's routine.
Yep.
Sure.
So what's the number to get really good at something? You have to do it for 30,000 hours?
Something like that.
Yeah, I think it's not that bad. It's like 10,000 or
I thought
something
it was
like
10,000 hours.
Yeah. Okay. Kathy, I think you're next, right?
Okay, I'm puzzled by this one. I'm wondering if it's a
joke?
no, whether it's for TWiP, but I'll read it. Tony writes, "Hello, Parisians. Here's a tumbler you might enjoy. What I learned as a field biologist. Humorous anecdotes from the world of doing research in the jungles of the world. Here's a sampler from the longer entry here." It gives a link. "You may then absent-mindedly scratch some of the 200-plus seed tick bites you have all over your body from 3 days prior, which it took 2+ hours to pick off with a pair of tweezers because you're too hairy to use duct tape. In scratching said bites, you may then unknowingly transfer hookworm eggs, newly acquired from the aforementioned jaguar poop, from your fingernails into your skin at the bite sites. You may then notice that some of your bites turn funny looking and start to actually travel across your skin and begin to itch so profoundly that it feels like your bones are burning, all because these particular hookworms can't get through the human dermis. Enjoy. It's a good laugh, if nothing else. Keep up the good work, guys." So I didn't get the Parisians reference.
That is a good description of larva migrans.
Parasites.
Yeah, okay.
There you go.
Parasites are not
He
people
did
from...
send it to TWiV. That's why I put it here.
What's
but-
interesting is that the egg itself will not do this. It has to actually hatch, and then it has to sit in the soil for about 3 days before it evolves into the stage that's infectious.
Mm.
And at that point, then you can just put your hand down, and they will penetrate through the skin, but they won't continue because they don't recognize us as their definitive host. Then you get something called creeping eruption.
Yep.
So,
Well,
from
yeah,
jaguar
I think
poop,
you're right,
eh?
Kathy. You're right, Kathy.
It was from the TWiP.
So it's a preview of next week's TWiP. Okay.
There you go. We can do it like that.
Who's next? Alan?
I think I might be next.
All right.
All right, so Michael writes, "Hello again, TWiVites. Hello, Dr. Racaniello. Hello, Dickson. Hello, Rich. Hello, Alan. Hello to any guests that may be present." All right. Kathy, I guess you're going to start appearing on these lists shortly, I'm sure.
Well, yes, because he says he's only on episode 175, so...
Right, so he hasn't gotten up to you yet. And he says, "It's been a while since I've written into the show. I became a regular listener in the late fall of last year and decided to try to catch up on all of the previous episodes before I wrote in again, but I just cannot help myself. I'm only on episode 175 because my Microbe World app stopped working over 3 months, and as a consequence, I'm just now catching up on the episodes from this year. I wanted to wait to write this email in case you guys ended up discussing this or something similar on one of your shows, but I can't help myself. I need to get this out of my head and into writing before I forget to send it. So, here goes." And he outlines this idea for combating influenza by genetically modifying domesticated swine and fowl worldwide to either underexpress or not express the sialic acid receptor on the surfaces of the respiratory epithelia via gene knockout. And Michael sees that, obviously, the goal is to eliminate their ability to spread influenza, and he sees there could be some problems with this. Specifically, the receptor might be performing an essential function. It might not be possible to selectively knock out just the sialic acid receptor. Knocking it out might force the virus to evolve affinity for a different receptor. And this might not affect the spread of the virus through wild swine and birds. The scale might be too big to put it into play. And, of course, he points out the anti-GMO crowd would not be happy about this. Genetically modifying all of the pigs and fowl that are used for farming. I was wondering if you guys think that this could be done, if only in theory. Please tell me your thoughts.
If only in theory.
Then it's not done, right?
Right.
That's kind of like grad student proposition exams, where they can do almost anything in theory, no matter how expensive
Yes.
or how many graduate student hours it would take to do it.
That's right.
Yeah, in organic chemistry, you assume that on the exam, you assume that all steps have 100% yields and the reagents are free.
That's right. That's right.
In theory, sure. In practice, I would say no way.
Right.
I mean, sialic acid's there for a reason, so I'm sure you can't get rid of it or even reduce it. But
Yeah, I think that's the biggest problem.
Yeah.
Is my guess is that if you knock this down or try and change it around, the pigs are going to be sick.
you could never replace all the pigs and birds with your modified ones, right?
Hardly.
Right.
So
Hardly.
that's the problem. You can't do it.
Nice
And
analysis, though.
Yeah, it's
Interesting
good. It is
idea.
good
It's got all the
analysis.
caveats right.
It's good.
Yep.
I'm glad you got it out of your head, but don't wait next time. You can do it right
That's
away.
right.
Yeah,
That's right.
yeah, you can toss ideas at us.
We don't mind if we've talked about them because we like to revisit.
Exactly.
Fine. Let's do just, should we do one more here?
Sure.
Move on to, this is from L. Absolutely love your show. It has renewed my love for this particular sector of science and inspired me to reach for a career in microbiology and immunology. Please do not stop what you're doing. There is a lack of educational yet exciting medium out there. If we had more people like you in the world, Honey Boo Boo wouldn't have left the cutting room floor. I don't get that, do you?
It's
Some
Go ahead,
reality.
Alan.
I know I haven't watched it, but everybody's referring to it, and I guess it's some.
I looked it up. It's a reality television program on TLC that features child beauty pageant participant Alana Honey Boo Boo Thompson along with her mother June, her father, and 3 older sisters. So it should have been on the cutting room floor.
Oh, yes.
At least for us, anyway.
Indeed.
family too. Okay, yeah, that could have, yeah.
Recently, there has been a ruling in Canada. She sends a link to an article. HIV disclosure ruling. The Supreme Court of Canada has ruled that some people with low levels of HIV who use condoms during sex do not need to disclose their condition to sexual partners. Wondering what your thoughts are on this issue. In a previous show in 2011, you touched base on how low variance loads are virtually undetectable by standardized tests when given to proper treatment. However, I was curious if immunosuppressed individuals have an increased risk of exposure. And with the evolving rate of viruses, what is the risk factor of an individual no longer responding to antiretros like HAART even after taking it for many years? P.S. Thanks for expanding my collection of books that my poorly put together IKEA bookcase can no longer support.
I'd get a Kindle, Ellie.
Yes.
Truly. I think this ruling is crazy.
I do too.
I think it's absolutely crazy because she's right. If you have an immunosuppressed partner, they could
Sure.
be infected at loads that a non-immunosuppressed person wouldn't be, right?
Right.
Right.
So the first line of the news thing is the Supreme Court of Canada has ruled that some people with low levels of HIV who use condoms during sex do not need to disclose their condition to sexual partners. But then there's someone at the end who says she thought that Friday's ruling was a step forward at first, but on closer examination, she said the ruling could make her criminal. I can still have a vindictive lover say that I did or didn't use a condom. It
True.
still becomes he said, she said.
He sheds.
Yeah, that's right.
I like that one better.
Oops. Yeah.
It's a good title. He sheds.
She sheds.
Excellent.
It's true.
This overturned an earlier decision that if you didn't reveal your status, was a crime.
What's the law in the U.S.?
No idea.
I don't
I
know.
don't know.
I don't know what the law is.
Because I'm not. I mean, this to me, this seems like this is a really thorny issue.
Yeah.
Yeah.
I mean, because the charge, it's not just that legally you're supposed to disclose it. It's that if you don't disclose it, it's what is it? Aggravated sexual assault.
Yeah.
Sexual assault and aggravated assault. Yeah.
Yeah, which can land you in jail for life. And that, I don't know. That seems really extreme and probably not in the best interests of public health.
According to the web, for the most part, the decision of whether to disclose HIV status rests in the hands of the infected individual. There are some circumstances in which it is legal for others to disclose, but that's just disclosure. That doesn't say about
Yeah, and I know there have been cases in the U.S. where people who have maliciously spread the virus have been convicted.
Yeah, that's
And-
Right, that
that's-
makes sense. We did one of those on
yeah.
TWiV.
CSI Virology, the most popular episode of all time.
Really?
Is that right?
Yeah, the most downloaded.
I'll be darned.
Okay.
Yep.
Well, the lawyer for this client says, what you're talking about here is a vulnerable, marginalized group of people who are going to be forced to go around volunteering to anyone with whom they're going to have sexual contact that they belong to that vulnerable, marginalized group.
Yes.
So it is
That's-
pretty thorny.
I can understand that nobody was going to want to sleep with you if you tell them, I got low levels of HIV in my blood, right?
Yeah. And his group's position is that a low viral load or the use of condoms should be the required test to avoid being prosecuted, but not both, as the top court said in Friday's ruling.
Yeah. Now, my argument would be that somebody with HIV ethically, I would think they do have a responsibility to tell their partners this. And if somebody with HIV asked me what they should do, I would tell them that.
Yeah.
But my problem with this situation is getting the law involved in such a draconian way. That doesn't seem productive.
Yeah. But, condoms are not infallible and
True.
Right.
still be infected and so
Right,
forth.
sure. Which is why I would advocate that somebody with HIV should disclose this.
Yeah, yeah, for sure. I would think
But
so.
unless it's being done with malice, I don't know about going after him for aggravated assault.
It's like it's just too bad that we have to have the law involved in this. I
Yeah.
agree with you, Alan, that you sort of have a moral, social, ethical responsibility.
Well, it's not just HIV, right? It could be any infection that you have. So, yeah, it's an interesting problem, isn't it?
Mm-hmm. It's complicated.
Should get a lawyer on to talk about it.
Yes.
Yeah.
Okay, let's do some picks of the week. Rich.
I have just been itching to do this for the longest time. Hang on, my phone's ringing. I got to hang it up. There we go.
Oh, you can take the call. Go ahead.
Yeah, I took care of it.
It's
So-
your plumber. Take it.
My pick of the week is barely science. It's the Vendée Globe, which is a single-handed, non-stop, unassisted, around-the-world sailboat race.
Woof.
And unfortunately, I've been away for so long. This thing started 27 days ago, but that's okay. You still got time to get on board, as it were, because it lasts for 3 months. Jeez. And there's a wonderful website for this. My justification for making this a science pick is it's very tech heavy. Okay, and the technical aspects are as interesting as anything else. There's a terrific website for this where you can follow the whole thing. These guys have all got amazing communications on their boats. They take videos, there's constant blogs, and there's a website that tracks the race. And importantly, you need to go under and select out the tracking, and then under the tracking, click the little link that allows you to that shows you where the wind is going. Okay, so that adds to the strategy of the whole thing. So these guys leave from France, sail south through the Atlantic, and then eastward, circling Antarctica by Cape of Good Hope.
And a
ultimately
tiny little
Cape
boat?
Well, not
Yeah.
tiny. They're 60 feet long.
60?
60 feet long. And
That's pretty
they're
cool.
special designs for these boats because they've got huge sterns, and they're very pointed bows, and they have a very shallow draft. These guys are in the Southern Ocean where they're doing like 20-foot waves and 30-knot winds, and the boats are designed so that they surf down the face of these waves.
Wow.
And these guys are sleeping under these conditions.
They're called sleds.
It's just incredible.
Wow.
The whole thing is incredible. So I'm addicted, all right?
Got it.
And I follow every day. And it's like following any sporting event. You get to know the skippers, and you follow the positions and that kind of stuff. And what they are enduring is just fantastic.
I bet.
I recommend I recommend getting addicted to this.
There's a little thing that tells you where they are right now. They're at the Cape of Good Hope.
The tracking link is the most interesting. Okay, if you go under.
Holy cow!
Yeah, and then you can do the show where it's night and day by clicking on the sun logo, and you can see where there's an iceberg that's been detected.
Yeah. So if you're looking at the tracking link now, they've got these. They have course markers that they have to navigate in a certain way, and so there's this one gate they're calling that the leaders are at called Crozet, which is 2 GPS markers in the Atlantic, and as they're sailing the race, they have to cross north of that at some point on their transit, and a whole bunch of them have sailed south of that because that's where the wind is, and there's a light wind right around this thing. But so there's a whole slug of them that are now heading north to just try and barely get the tip of the thing.
You can hear these. You can do live.
Yeah, there you go.
This guy's talking live right now. He's in this way down there, south of Africa.
Yep.
Wow. He's in French.
So they have they post daily video summaries that are translated into English, and then there'll be a weekly summary that they post that's a longer video that they translate into English. So there's nice summaries, but you get.
It's cool.
It started with 20 boats. 7 have already dropped out for various reasons. Getting one woman. There was one woman in the race. I was sorry to see her go, but she got dismasted in the North Atlantic.
Dismasted.
And some other guy lost his keel. 2 people ran into trawlers in the middle of the race. Some other guy ran into a harbor buoy that was loose in the mid-Atlantic. It was too bad. He was a great racer.
Well,
and you think science careers are hard?
I bet you they can hit a whale every now and then too if they don't watch it.
Yeah,
There's an iceberg here.
Yeah, this is an amazing race.
Cool.
So get hooked.
No, I don't think so.
Get hooked vicariously. That's what I mean.
Yeah, me too.
I'm not doing this.
You're not going to do it, but you're going to watch it, though, right?
Yeah, you bet.
Wow.
Cool. That's pretty neat. Alan.
I have a related pick. When I saw Rich had picked this, I said, now is the time to do this. This is a video of the record-setting circumnavigation that just recently took place. It was the 2011-12 run for the Trophée Jules Verne. The Jules Verne Trophy is for sailing around the world nonstop. You leave Cape Horn to port. So it's pretty much the same route that Rich just described. It goes from France, it goes down to the Southern Ocean, around past the Cape of Good Hope, Cape Horn, and then back up to France. This, however, is not a single-handed crew. This is sort of the unlimited class version of that. And the video is the most recent winner of this and world record setter. It's a French team sponsored by Banque Populaire, and so that's the name of their boat. They finished in 45 days, 13 hours and change.
Oh, wow. I mean, that's like twice as fast as this single-handed race.
This thing and this video, I suggest looking at it on a desktop machine, setting the resolution to HD, and put it to full screen. It is spectacular.
Huh.
This boat, and every now and then they'll show a little glimpse of the knot meter.
Huh.
And you see that this sailboat, this trimaran, this big trimaran, is doing 35 knots.
My God.
And then they pan to the horizon with the icebergs on it, at night.
We had another boat go down like that, actually.
It's just crazy. And these, not only, this boat is very, very lightly built because, of course, they want it to go as fast as possible. So they're bawling through the Southern Ocean past these ice fields, and stuff is breaking, and they're molding a new fiberglass patch down in the hull so that they can put it on the mast, and some guy's climbing up the mast, and the waves are rolling along behind them. It's just absolutely spectacular.
Wow.
Incredible amount of technology in this boat.
I've only started this video, but it does look incredible. Right. As soon as we're done, I'm going to finish it.
Yeah, and 35 knots is that's 40 some odd miles per hour, and you can convert to metric if you like. But it would be at speed. It would be hard to catch that boat in most motorboats.
Wow.
Yeah, in any boat, that's
They're
crazy.
booking.
I mean, when we go 6 knots, we're scared to death.
Yeah.
And these single-handed boats go routinely 15 knots and sometimes up to 20, but 35 is just crazy.
The most important advice that they give new trainees on these boats, apparently, is always sleep with your feet forward.
40 miles per hour.
If you hit something at that speed, you don't want your head going into the bulkhead.
No.
Yeah.
Goodness.
Okay, Kathy.
So I picked science cookies for something completely different, but
These fun.
it is just in time for the holidays. And so the first one you'll see is a Petri plate cookies. Those might be about the easiest to do, but they've got the gel electrophoresis squares are quite elaborate. These people have way more spare time than I do. But we got these as part of the link to our departmental cookie exchange, which is coming up for those who want to participate. And I just had to share it.
I think these are terrific. I really cracked up when I got to the lab mouse one.
Yes. Yeah.
It's got one mouse that's dead.
Yeah, that's great. That's very good.
Yeah. Drosophila. There's also science cookies.
Funny.
Yeah, it's just zebrafish, atoms. Yeah.
I remind everyone when I was at the Harvard virology retreat last year, they had They had a contest to bake virus stuff, cookies, cakes. So I put pictures of those on Facebook.com/thisweekinvirology.
I remember
Great.
that. Yeah.
Check those out.
Ha.
Dixon, do you have a pick?
I do. It's another NASA pick. I picked NASA last time, so I want to pick one now. They have put together a series of high-resolution night photographs of the Earth, as seen from an orbiter satellite that's in a geosynchronous orbit of the Earth, so they can get long exposures of the same area at night. And you should go to their website. It's up on their website right now. It's quite amazing to look back at the Earth and to realize that you can see where you live, even if the sun isn't shining. To look at the United States at night, and you can see every light. It really reminds you of the fact that we are an energy-driven society and that we need to cut down on that somehow.
You could see every light. I could see my house.
You can see in Western Massachusetts. I know we can see your house. It's amazing. It's quite amazing. Actually.
It is. Yeah, the resolution on this thing is very impressive.
It's remarkable. Even though that picture's been shown before, this is now at new high definition resolution.
Yes.
So.
Wow.
And
Worth
it's from a
mentioning.
new time. It was
It's from a new
astronomy
time.
picture of the day.
See that?
Picture one of the days this week.
There you go.
Yeah.
Dixon, is the Earth living?
Listen, it's Gaia. Of course it's living.
Are there other living planets in the universe?
There must be, Vince. There just must be there. The odds are in favor of
Us
it.
astronomers have that argument, and
Of
we have
course
the
they do.
living, non-living argument.
Well, yeah, that's true.
My pick is an artist I found yesterday. Trilobite Glassworks. Did I get that right, Dixon?
You did. Trilobite. That's right.
No, Trilobite.
No, trilobite.
Trilobite.
I
Well, that's what I said this morning, and you said I was wrong.
No, you didn't say it that way, Vince. You
I said
it-
trilobite.
You did. You said Trilo. So trilobite means it's got 3 lobes. That's what it means.
Trilobite?
Yeah, trilobite.
Trilobite?
Trilobite.
Anyway, this is an artist who makes things out of glass and all sorts of things, but a lot of viruses and bacteria and eukaryotes.
Yes.
I'm looking at a variety of viruses: Ebola, Euglena, different microbes, Ebola virus. They are really cool. She makes
They're very nice.
magnets.
Yeah.
She makes brooches and other things.
Pendants. Yeah.
Pendants.
She says for the pick a virus, if you click on that picture, made to order fused glass magnet brooch or pendant, allow 4 weeks. Do you have a favorite virus? Are you partial to HIV or herpes or dengue? Perhaps Ebola is your favorite. I will design and create a fused glass magnet brooch or pendant with your chosen virus. Please contact me for feasibility.
Feasibility.
Right.
The phage is beautiful. I
Yeah.
love the phage, but it sold out yesterday
Oh, well.
to another microbiologist because he was making fun of me on Twitter yesterday. He said, I bought it before you.
Oh, brother.
I'd like
Collect
to
them
have,
all.
They're $30 each. That would add up, but
It would.
I ordered a couple yesterday. I think they're just so cool looking. They're
I think
just
it's fascinating that an artist takes, has these science themes, in particular microbiology.
Yeah.
Well, you said she was a scientist at
She
one
said she
point.
used to work on paleontology. That's why the name
okay.
is trilobites. Trilobites?
No,
Trilobite.
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 Accent on the try.
Trilobite.
Yes, try.
You got it.
Isn't it tribbles? Tribbles, right? Trouble with tribbles.
Now, we're
Trouble.
probably going to get letters from paleontologists saying that, no, it's true.
Is the
All right.
page still on the site? Because I didn't see it.
Yeah, if you
Go
go
away.
to that pick a virus made to order,
Uh-huh.
and there's a little icon in the upper right.
Okay.
If you click on it, you can see the picture there. Should probably make another one, but
That's a
Oh, I see
complicated
it. Yep. Okay.
it's a refrigerator magnet. She says, keep the microbes on your fridge, not in them. That's great. She's wonderful. And of course, this is Etsy, and Michelle Banks is also on Etsy, who makes watercolors of viruses too. So collecting all these microbial artists. So there you go. And we have 2 listener picks, which we've mentioned. One is Ken's Spanish flu simulator and the communications programs of Stephen, along with Sherp. And that will bring us to the end. We do this every week and release the episodes on the weekend. You can find them at iTunes, TWiV.tv, and microbeworld.org/TWiV. And our Facebook page has all kinds of neat stuff, facebook.com/thisweekinvirology. Do send your questions and comments to us to twiv@twiv.tv. Dickson Despommier, thank you for joining us again.
My pleasure.
Dickson's at medicalecology.com and also many other places. Rich Condit, welcome back.
Boy, it's great to be here.
Missed you, and we're glad you're back.
Yep.
So am I.
Poxdoc.com, is that right?
No, but that'll do.
Let's see who's at poxdoc.com.
I used to use Poxdoc as an instant message handle.
You should have Poxdoc. Go buy it right now before everyone else does.
Poxdoc.com. Yeah, I
somebody's-
suppose I could, yeah.
Somebody's on it. Kudenkan Yudan Shakai of Virginia.
You're kidding.
Gone already,
But the site's not working. So
Huh.
Actually, a long time ago, I made you bit.ly/poxdoc. That should still work. Yeah, that goes to your University of Florida.
All right, cool.
At Gainesville, bit.ly.com/poxdoc.
I got to remember that myself.
Kathy Spindler is at the University of Michigan. Thank you, Kathy.
Thank you for having me. As
Always.
always, it was fun, and I learned a lot.
Always a pleasure. Now,
That's
who
that's
says
Alan.
That? Who says that?
Gee, I-
my line.
Alan Dove is at alandove.com and Alan Dove on Twitter. Thank you, Alan.
You're welcome. It's pleasurable.
He's not going to say it. He's not going to
Come on,
say it.
Alan.
You got to say it.
Do it.
It's always a pleasure.
There you go.
You get email if you don't say it.
That's right.
I'm Vincent Racaniello, and you can find me at virology.ws. You can't actually find me, but my writing,
You're close.
all the podcasts are there, other things. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. Now we have some great candidates for titles here.
Uh-oh.
Oh, I like this.
I love Liver Let Die. or Bond, Covalent Bond.
Covalent bond. Yeah, I love
You like
that.
that one?
Yeah, I like that a lot.
Yeah.
Yeah, let's do that one.
Yeah, okay, we'll do Bond. But Liver Let Die is great.
It's
Yeah,
true.
it's good.
And Dickson had a good one, too.
He did.
He had
Nope,
50,
another hepatitis thing somewhere.
50 ways to leave your liver. That was good, Dixon. A shrewd approach to receptor identity. Ah, that's good, too. Hey, these guys are all clever. That's why you're on TWiV.